Hello everyone and welcome to today's webinar in our COVID series, where we're joined by two speakers, the returning Professor Ray Kruse Iles and new speaker Professor Akhilesh Pandey on their work in this area. So now I'll hand over to the first of today's speakers, Professor Ray Kruse Iles from MAP Sciences and the Department of Veterinary Medicine at the University of Cambridge. He's the Chief Scientific Officer at MAP Sciences and founder and Chief Scientific Officer of NISAD Lund. Professor Iles has been head of the Williamson Laboratory for Molecular Oncology at St Bart's Hospital in London, and was recently the founding dean at Abu Dhabi's University College of Health Sciences. Recently, he was appointed visiting research professor at the University of Cambridge, and today he's going to talk to us about the development of a clinical MALDI-ToF mass spectrometry assay for SARS-CoV-2, its rational design and multidisciplinary teamwork. So it's over to you, Ray, if you want to take control.
Thank you very much. I'm just trying to take control now. It's being a little bit slow again. I'll keep trying and hopefully I'll get my first slide up. Dan, I'm afraid you're going to have to move it for me. Sorry. Let's have and I'll try and take control again. Thank you very much. And thank you very much for allowing me to come back and have another shot at this to give you an update. My first three slides are going to be a sort of recap on the last talk, but we've moved on tremendously. And we've been joined by an awful lot of people in the UK and in the US, and of course the manufacturer of one of the mass spectrometers. And you can see how all this fits together in terms of this rational design, this multidisciplinary teamwork. And I thank the team enormously for what they've done. Just see if that works. Let's try that. Oh, sorry, it is working. Let me go back one. Will allow me to go back. The first thing is to remind you that you're probably familiar with MALDI-ToF mass spectrometry being used clinically for the detection of bacteria and the identification of bacterial species. Now, in that case, everything has been optimized to look for housekeeping genes within the bacteria, so ribosomes and other internal proteins that tend not to be glycosylated. They tend also to be small, and they're easily ionized by CHCA, so they're easily fly molecules. But when we look at the virus, those housekeeping genes on these housekeeping proteins are not present and are not a feature of the viral infection. So we have to look and optimize for proteins that are characteristic of the virus. And if we look at the virus itself, the main features are a host of very large glycoproteins, which are embedded within the envelope of this envelope virus. And of course, what we want to target is specific proteins, such as the spike protein, which are unique to the SARS family and hopefully for COVID-19. So there was a lot of work that had to be done to optimize to look for large glycoproteins. So the matrix, the conditions in which you conducted the mass spectrometry were completely different to what you would be using to look for bacteria. One of the things that we also realized is that sensitivity was going to be an issue. And what we have done is worked out a very simple and, if I'm allowed to say the word, cheap method to concentrate the virus within the biological sample, so that you can get a very concentrated sample of enriched viral particle, which you can then put onto the mass spectrometer. And this is actually very easily achieved. You add ice cold acetone at a 1-to-1 volume with your biological fluid or biological sample that you've got. You spin at 16,000 G at 4 degrees for 30 minutes, and you precipitate and therefore enrich the precipitate with your viral particle. You leave an awful lot of proteins still in solution if you're using just a 1-to-1 ratio of acetone at this temperature and for this time period. You do bring down other very large proteins that might be in that biological fluid, which will appear in your mass spectra, but sometimes, as this is proved in the development of this test, this is actually very useful. The next stage that we had to overcome was the release of these large glycoproteins from the viral envelope. We achieved that by empirically working through an awful lot of chemicals which were able to disrupt this viral membrane. And we found mass spec grade, mass spectrometry friendly dissolution buffers, which didn't suppress ionization and didn't interrupt or disrupt the mass spectra which we were able to generate. Furthermore, it was able to break apart the subunits, and if we added dithiothiazole, which is a key component of this dissolution buffer, we were able to break the disulfide bonds which held many of these subunits together, because often they are cleaved to activate them, and we were able to get cleavage fragments of, for example, the spike protein, the S2A, S2B and S1 fragments, which were of the right mass spec, right molecular weight to be seen on the mass spec. So the dissolution buffer that was developed is absolutely a key component in getting this test developed. Now, to get all this working, we couldn't really be initially playing around with live virus. So luckily our colleagues at Cambridge and at University of Kent had developed lentiviruses, which were lentivirus pseudotypes, which were able to express the large glycoprotein, the S protein complex, from the SARS coronaviruses into their membrane. Now, these pseudoviruses were fantastic for us to play around with and get all the conditions right with, because here was our virus construct. It had many of the envelope proteins, but the core was inert. It didn't have the viral nucleic acid, so it wasn't dangerous, couldn't be replicated. But we had a very good mock version of what we were trying to do, which was to harvest and look for the viral proteins. So everything that we developed previously could be modeled on these pseudotypes. And this is the kind of spectra we were getting. This is the S1 protein or fragment from the spike protein, very clear spectra, close to 80,000 Daltons, and a host of large viral envelope proteins which are coming up from the lentivirus pseudotype that have been developed at Kent and at Cambridge. We were then able to move on to testing the optimized system on live virus, and Professor Heaney's group were able to get SARS-CoV live virus, 2 different isolates. They were able to grow them in culture, and we were able to take the culture media and look to see if we were getting a similar kind of spectra. And yes, we were. There's our S1 protein, and also a host of viral envelope proteins as well. We then moved on to looking for the virus in gargle sample, because this seemed to be the most optimum medium or sampling technique that we could have, where people could take a gargle sample at home. Literally, they just take 10 mils, and it turns out that water is the best. So our sampling solution is take a 10 mil sample of water, give a very good gargle for 30 seconds, spit it into a tube, and then send it to the lab. We would then do the preparation stages that I've already shown, and we could do that in batches, which would take about 45 minutes, 46 samples we can run at any one time. And the machine would be churning out results every 3 minutes. And we obviously had our reference spectra, where we got these large envelope viruses, our S1 fragment. We do get a problem if we're growing in culture, because we get BSA interfering, but the S2B is under here in this region, but you can see that. When we started to look at gargle samples, yes, there was the S1, there were the large viral envelope proteins, which we started to characterize by molecular weights and called them VEPs, so VEP 26 to 20, sorry, I can't even read that, to 28, etc. So we could characterize the viral envelope proteins coming up as well. Now, I did mention that depending on the medium that your viral sample was in, you also got other large proteins coming out in your preparation step. So if they were big, they would also be co-precipitated in the acetone prep, and they would also show up in your spectra. Well, actually what was happening is that saliva and the wash of the mucosal layer in the throat was picking up immunoglobulin A, which is about 330,000 Daltons as a dimer with its S chain and J chain. And when we put in our dissolution buffer, it would break the immunoglobulins into heavy and light chains. So we were also picking up in our saliva samples not only whether there was viral envelope proteins, which could come from a host of viruses, the specific marker proteins which told us that it was COVID-19, we're also picking up the humoral IgA response as well. There was a host of other small molecules which are being investigated, but that's another story. We started to see how effective this methodology was, or is in fact, at picking up these viral envelope proteins. So we were able to get negative controls, 2 people who were negative controls, and we got them to give us 40 samples each, and we analysed to see if they have S1, S2, the VEPs. And we also have spiked live virus into the control COVID-19 negative, PCR negative samples, and ran it as if it was a real mock sample. And there we can see huge signal intensity coming up from the S1 peak. S2 peak we couldn't really show because the culture media we were spiking in had albumin, so it could obscure this peak, so we didn't bother to measure it. And we also started collecting from unscreened volunteers, some of which had reported symptoms, but we hadn't got PCR data on there. And the big problem we have is that there is no refrigerator in the world where people have taken swab samples and simultaneously taken a 10 ml gargle sample for us to compare. So we're a little bit stuck, but we did start to pick up S1 in some of these people. We started to decide what was cut off and grey. This person in particular had just been recovering from a COVID-19 probable infection because I had all the symptoms and was laid low for a good 2 weeks in bed. And that person in particular, we saw elevated S1, S2 peaks, S2 A as well, not as high, and then all the viral envelope proteins. But these aren't specific, they could arise from a multitude of different viruses. So everything is looking quite good, and that the S1 in particular seemed to be our primary marker for saying whether you got COVID-19. An added bonus was looking at the immunogoblins, which were also coming out in this gargle sample and were also visualized on the mass spectra. Of the COVID-19 PCR negatives, one had reported a sore throat, the other one didn't. Spot the one with a sore throat. And we realized that there seemed to be some correlation between infection and the level of immunoglobulin light chains and immunoglobulin heavy chains in our cohorts which we're looking at. So these are our COVID-19 negative, repeated 40 times. The IgA heavy chain was the most interesting feature that we picked up because we had anecdotally noticed that high levels of IgA heavy chains seemed to be correlating when we got in our volunteers where we picked up VEPs. So when we were picking up the other viral envelope proteins, whether they were the spike protein or any of the other viral envelope proteins that seem to be detected in our mass spec profile. Now, the IgA light chains didn't seem to, or didn't, in fact, correlate with any of the VEPs, but the IgA heavy chain did indeed, the levels of the IgA heavy chain did indeed correlate with whether you were able to detect VEP, and correlated with the levels of the other viral envelope proteins. So it looks like we've got quite a good test that's going to give us an awful lot of clinical information. We can look for the corona-specific proteins, and we can look for viral envelope proteins for a general viral infection that you might have in the oral upper respiratory tract. We can look for the immune response. We're building software. The cutoff levels are sort of being determined, but basically we could have a green, they're low, grey, oh, maybe we should repeat your sample in a couple of days' time, or red, because we consider it to be elevated, that you're in a definite high positive level, and also what your immune responses are. So this is what we started to develop, and that's sort of what I've got up to and published or submitted for publication. But there's a very big difference between having this initial great response and actually producing a high throughput test which you could run on millions. And this is what the team have been doing for the last month or so, and I'm just going to give you the kind of data that we haven't even put out there yet, bringing you fully up to date of where we are in developing this test. So for high throughput testing, as you've probably seen from the UK government, if they want to do 10 million people a week, we've got to really push this out and get this whizzing through the mass spectrometer. So we push them until they break to find where the failure points are. Now, one of the features we do is 8,500 spectra, and we average that per sample. So that's an awful lot of shots on each sample. The reason is to give us good reproducible data of where we do a quantification. We're confident of the quantification of that signal intensity. We were running 300 samples today in an 8-hour shifts per day. And then we're identifying the failure points. Now, the biggest point of failure is actually with the instrument itself. Now, cinepinic acid, unlike CHCA, causes an awful lot of splatter because you have to hit it quite hard with your laser energy, and that splatter builds up onto the primary lens. That's also complicated and can get worse when you've got other additives in there, such as our viral dissolution buffer. If you had SDS, that would completely wreck your instrument. So it's a balance of getting all these materials right, that they give you the result you want, but don't damage your instrument. But basically, this primary lens fouling is a major problem that you'll find in all your mass spectrometers. The easy solution is just to take that lens out and clean it. And it takes 2 minutes, a methanol on a cloth, and you can clean this lens. Now, this is the lens from the 8020, which did have, because fouling of the primary lens is known by the manufacturers, a laser ablation, which was designed to clean the primary lens. And they had done this after build-up of CHCA. But as you can see, cinepinic acid and with the buffer, that cleaning protocol with the laser ablation didn't actually get everything, and critically, it wasn't able to get inside the bore of the primary lens, where the build-up, when it occurs, starts to detract from your mass spectrometry quality and the intensities of what you can detect. So the easy solution, and this is quite good on the 8020, is to take the primary lens out, clean it regularly and put it back. What we've done is set up several QCs to look at run performance, also check that every part of the system is working, because there are other failure points that you can have if you're trying to run a high throughput screening system. So this is a major point that people have to consider if they want to try this on their mass spectrometer. How easy is it get to that primary lens and clean it? So not all of them are that easy to get access to this primary lens. I'm just going to give some examples of some of the internal QCs we have. Interestingly, while we're running all this, let's break the machine, let's do high throughput, is that we found some people don't gargle. They don't know how to gargle, and they can give us a full sample. But we can tell whether it's a full sample. If someone was to take the water and just sort of swill it around their throat, this is the kind of spectral line we get. So we can tell if it's a full sample, and we can say, no, resample, you haven't gargled properly. When you do gargle, you get these interesting proteins that do come up. This one here is one of our key markers. It's extremely constant within the gargle. And if we don't see that, then we know that they haven't taken the sample correctly, and we immediately reject the sample and have to ask them to resample. The QC peaks, we tend to look at the very high mass peaks. So we run, instead of live viruses as a quality control, we run pseudoviruses and look for the S1 protein. But you can measure the intensity of this, and just to give you an idea how fouling can cause deterioration in your measurement. So we have worked out on our instruments at which point they are failing, or that we cannot rely on the results because that fouling is reducing the reliability and the intensity readings that we're getting from peaks. So these QCs are extremely important if you're going to take a system out for high throughput mass screening. Having known all of this and having all our quality controls put in place, we started to get our systems up ready to pass validation trials. So particularly for the Food and Drugs Administration EUA emergency use, we want to make sure that we can meet the requirements there. So the first thing we started to look at is limit of detection, and we can do that by in vitro samples, for example, spiking live virus into the biological medium, which is obviously this gargle. So what we did was take the COVID-19 virus grown in culture. It was filtered and spiked into culture media, into the gargle. The team at Cambridge determined the biological activity, our plaque forming units for that virus, but they also were ready to measure the PCR copy numbers in the mock samples that we created. So we got a PCR result. We knew what the PFU results for the amount of virus we've added to the gargle solutions, and we measured the intensity of that key marker, the S1 peak, at 79,000 Thompsons if you're using the old language, or MZ, whatever you prefer. So we were able to do a direct test of sensitivity, and we were working at limited detection. Well, to be honest, we took it at the limit of quantification. So we were looking to see at what point we got a clear, distinguishing, quantifiable intense peak above the baseline. So here about 50% of samples were above the baseline. Here all of the samples are above the baseline, so we're taking that as our limit of quantification. And that wasn't too bad, because we were comparing against PCR tests which have already got approval. Some of them are absolutely incredibly sensitive, claiming to be down at 40 copies per mil. Some of them are a lot higher, in the regions of several thousand copies per mil, which is their limit of detection. And the results were that we were not as sensitive as this very sensitive RT-PCR method that had been developed by Professor Heaney's group, but we were comparable. We were pretty good at picking up in terms of plaque-forming biologically active virus, which equated to about 75,000 copy numbers, which is comparable to about 25% of the PCR tests are out there. For example, the Luminex area system, we are measuring about the same level of copy numbers as that PCR method. So all well and good, we're getting there. But when you're doing validation, what you really want to do is see how it performs in the field on live patients. So we've got 2 studies underway in the UK, both prospective. One using the Shimatsu 8020, and thank God for our USA colleagues out at Northern Illinois University. I know Beth's listening. She's doing a prospective study out there. She's using a slightly different mass spectrometer, the axoma, and I'm going to give some details about that. Now, we have a big problem in that so far, things have not gone too well. We haven't got our simultaneous PCR sampling, so we've screened 160. We think we've picked up 3, but they weren't PCR confirmed at the time. We've got screen negatives. There is a difference, and I'm going to go into this sort of grey area of detection in a minute. But in the USA, they've been absolutely fantastic. They've simultaneously doing PCR on all samples and then doing the gargle test, the MALDI test. So they're reproducing the result out there. Now, at the moment, they're using the axoma MALDI-ToF, not the 8020 that we do. Now, this is bigger and it's slower. We're small and fast. They've got a longer flight tube. They use a nitrogen gas laser instead of a YAG laser. We have easy access to the primary lens. They have incredible difficult access to the primary lens. It's about a 2-day downtime. However, their primary lens, we figured, shouldn't foul as frequently as it does in the 8020, because they have a 2-chamber vacuum system, and they should be able to pull some of that splatter away because they have a primary vacuum in the sample bay, pulling away where the sample is placed in and fired on by the laser. I hope Beth forgives me because we've broken their axoma. After about a week or so, we've said stop. The quality controls have dropped so tremendously, we can't rely on the data. Delano and the Shimatsu team are out there repairing it and also getting them an 8020. But the 8020, after 2 or 3 plates, we just simply take the side off, access the primary lens and clean it, and within 2 hours we're back up and running. So, you know, we have to have figured out preventive maintenance for high throughput and running our 3-stage quality control monitoring. Okay, so just to give you an example how the axoma and the 8020 compare, there's some calibration and some focus differences, but this is our key peak, and we are picking up the same proteins. I won't talk about the Galliard peak, that's for another talk. The axoma, we are picking up all asymptomatics at the moment, and this is the kind of data that the axoma is producing. This is the PCR positives and negatives. When we look at it on ours, we do think we've got a grey zone, but because it's so quick and cheap to run this test, we think you can do multiple tests in any one to see if they're going from the grey zone down to green or into red. So there is a problem of overdiagnosis. You'll see lots of people being diagnosed as asymptomatic. Well, asymptomatic, what the hell does that mean? Is that they're pre-symptomatic or is it a false positive? And it turns out in studies that actually 80% of the asymptomatics that were picked up with some of these PCR tests are don't go on to develop symptomatic disease. Whether they are truly asymptomatic, we don't know. But regardless of this, I think what we have is a PCR complementary test with the MALDI-ToF technique that we're developing. So basically what we've got now is a gargle test. Split your sample in. We actually have a safety precaution where we put the sample pot in a UV 25 nanometer cabinet, 15 minutes to inactivate the virus but preserve our viral envelope. Filtration through to results, a lot of clinical information, but the main thing is that this is fast and it's cheap. if we're running the 80/20 at 100% capacity, the cost per test we've worked out is only 59 pence for the reagents. If you add in all the other costs, you're only talking about £2.48 per sample in terms of the cost that you have for running a sample. I'm rushing a little bit, and I've got an awful lot of acknowledgements here. The Map Sciences team, Raminta, Jason, Jonathan, Christian, Vicky, the HIT Consortium, Prof Jonathan Heaney's team, George, Alex, Mina, University of Kent, who developed these pseudoviruses, which are really important, is Nigel, and at Papworth we're getting a lot of samples from Helen and Samita. Best team at Northern Illinois University have been absolutely fantastic, and the Shimatsu team are providing fantastic support in the UK, in the US. The funders of this have been my partners at Map Sciences and NISAD, and the HIT Consortium is a recipient of UKRI grant. Just the last one, this was yesterday's data. I just can't resist showing data. This is a patient who was on a ventilator and has recovered. We can see the VETs and the S1, and this is a young person who we think is COVID-19 positive, elevated levels of IgA, the VEPs, and sorry, the IgA heavy chain and light chains. So you can see how we're trying to set the threshold, but we think we're pretty much there to get this rolled out. Thank you very much for listening.
Great talk, Ray. Thanks very much. There's quite a lot of questions coming on the Slack. I think we've only got time for a few of them right now, but perhaps you can go on later and follow up with any of the ones we don't manage to get to before we hand over to our second speaker to today. So I'm just going to pick a couple. Professor Kevin Mills at UCLH actually says, unfortunately at the moment there's a coronavirus doing the rounds that gives similar symptoms to SARS-CoV-2 in children. Can this test tell the difference between this innocuous strain of coronavirus from the COVID-19?
I don't know at the moment. We've got to see how different the S proteins are. I know seasonal coronavirus produces S1 spikes as well. There are other peaks in there that may be useful for that differentiation.
Great. And then one more quick one. Some other studies have shown the nuclear protein has higher levels than other proteins, but you don't detect it in your study. What do you think the reason for that is?
I agree with them. I'm not sure. We need to do more work. I think there's an awful lot of data there, and the nuclear protein might be there. It's one of those VEPs, but it's in terms of molecular mass, it might be very similar in all of the viruses.
Okay, great. And just because I'm curious, would you still expect to see the immunoglobulins present in someone who's recovered from COVID-19? And have you analysed any samples where they have recovered and had a positive antibody test to verify that situation?
What we have, what we show is that when we see the S1 peak, that they're extremely high, the immunoglobulin heavy chain and the light chains, but the IgH particularly high. And when we see the disappearance of the VETs and the S1, those levels drop tremendously, but they're still detectable.
And do you have any idea how long that might persist, or are you going to plan to try that?
That is one of the studies we're doing at Addenbrooke's, is to do all the follow-up on people who have been sick and see where these markers change. There's a lot more markers to come up as well.
Great. I think that's probably all we've got time for now, but please go on the Slack channel and answer some of the other questions.